In these slides, we'll talk about alternative splicing of messenger RNA. The idea of splicing and alternative splicing was given by Gilbert in 1978, and before this, this idea was the idea of split gene was given by Roberts and Sharp. This is very important because before it was said by the Beadle and Tatum that one gene code for a single protein. That was known as the one gene, one enzyme hypothesis, but afterwards, due to the discovery of this alternative splicing, it was known that more proteins can be made from a single gene. From single gene, more than one proteins could be made. The exons and introns of a particular gene get shuffled to create multiple isoforms of particular protein. Therefore, a single gene can produce more than one protein. More than 70% of the human protein coding genes are alternatively spliced. This is very interesting. More than 70% of human genes are producing more than one proteins. So there are different types of alternative splicing. For example, one is constitutive or normal alternative splicing, alternative donor site, alternative acceptor site, alternative position, and exon skipping and intron retention. So we'll talk about these different types one by one. So this is kind of the post-transcriptional modifications at 5' and there is the methylated guanosine attaches that is called the 5' and capping, and there is splicing. Introns are removed from the messenger RNA, and at 3' and there is addition of poly A tail. So what is the constitutive splicing? In this splicing, here you can see there are introns, there are 4 exons, and there are 2 introns. And these introns are removed, and the exons join together to make this coding DNA or coding messenger RNA molecule, which have only exons, and these are in the same order as these were present in the peri-mature messenger RNA, E1, E2, E3, E4. So this is constitutive splicing, normal splicing, where there is no shuffling and modifications of the exons. Similarly, if we talk about these peri-messenger RNA, we talk about the peri-messenger RNA molecules. There is the exon skipping. You will see that all these 2 introns and E2, these are lost in the splicing, and we can see these E1 and E3. So one exon is skipped, so that's why this is known as exon skipping splicing. The other is alternative donor site, so that is mostly at 5' and some part of the intron is taken up by the exon 1 at the 5' end of the gene, and this is actually this one is here you can see this is alternative donor site, and there is the some part of this intron is taken up by the exon. And here we can see this spliced messenger RNA molecule with a part of this intron. Similarly, at 3' and there is some alternative acceptor site. From here, some part of the intron is taken up by the exon 2, and that exon 2 is enlarged and spliced messenger and look like this. Similarly, in case of alternative position, some part of the exons are taken up by the intron, and some part of exons are spliced with the intron that leads to the formation of smaller messenger RNA molecule as compared to the original mature messenger RNA exons. Similarly, there is another type of the alternative splicing in which the intron is taken up by the whole intron is retained by the messenger RNA. The whole intron is retained by the messenger RNA. It looks like this. All of this is now changed into the coding sequence like exon 1. So these are different types of the exons and alternative different types of alternative splicing. I hope you like this video. Thank you very much.